Well, thank you so much for the invitation to speak. I'm just talking a bit about this case study identifying Brigatinib as a potential target for NF2 null tumors. And so it's very clear, if you look at a couple of reviews, that the complexity and involvement of Merlin in intercellular signaling is extremely complex, making it a real challenge to identify kind of the best or a really good potential target. So as part of the Synodus for NF2 consortium, and I will fully give credit to the whole team in my slide at the end. So supported by CTF, the Synodus for NF2 consortium involved a number of investigators, basic scientists, clinician scientists, to try and identify therapeutic targets in these NF2 deficient models. So a cell line screen was performed using almost 2,000 compounds in models of meningioma and schwannoma that were either NF2 proficient or deficient, with the goal of finding a specific sensitivity in cells that had lost Merlin function. And the plan was to progress the most promising combinations or agents to in vivo tests in both meningioma and schwannoma models, and in parallel and at the same time do some follow-up on the molecular responses using RNA sequencing to look at the effects on the transcriptome, and also a technique called MIB-MS to look at the kinome. And this is a way to look at the complete kinase signaling network en masse. And I'll talk a little bit more about that in just a moment. But the results of the screen really saw a few combinations rise to the top for two of these models. They showed a slight preference for increased efficacy in cell lines that had lost Merlin function versus a wild type. And the two combinations, interestingly, or two of the top three, I should say, in each of the types, had some overlap. And of interest listed here, a compound called ALK-in1 was identified in combination with an AKT inhibitor in the meningioma model, or with dasatinib, which is FDA approved for Ph-positive, Philadelphia chromosome-positive chronic myeloid leukemia. And ALK-in1 is actually a chemical analog of an FDA-approved inhibitor called Brigatinib. Dr. Plotkin and others have kind of mentioned this as it's currently under study in a clinical trial, but it was approved for non-small cell lung cancer that was resistant to crizotinib, approved in 2017. So this really represented also a potential drug that could be repurposed if it proved efficacious. And I'm going to show you the in vivo models first, a xenograft meningioma model, injection of BENMEN1 cells into the cranium of mice and treatment over 12 weeks. And the combination was tested in the meningioma model, and you can see, as indicated by the arrow, that the single agent Brigatinib in green was nearly as effective, or almost equally as effective, as the addition of the AKT inhibitor, shown in the purple line. And then at left, in a genetically engineered mouse model of schwannoma, you can see, indicated by the arrow, single agent Brigatinib was the most efficacious in reducing tumor volume, so DRG size, where these tumors form in the mouse. You can see a reduction in volume compared to vehicle treatment. So this is really exciting and really promising. The next approach we wanted to take, in addition to the transcriptomics, which I don't have time to talk about today, but something maybe you're less familiar with, is this approach called MIB-MS. It is a chemoproteomic approach to looking at the kinase signaling network, or the kinome, an attempt to kind of study all the kinases at once. Brigatinib is a kinase inhibitor, ALK is a kinase, and so the prediction was we might see targeting of ALK in these tumors and in these cell lines, and we wanted to investigate that using proteomics. And so we can get lysates from patient samples, we've done this before, or the cell lines or mouse models that we have, prepare a lysate and pass it over a mixture or multiplex inhibitor bead mix by gravity flow affinity chromatography. And so the MIB is the multiplex inhibitor beads, and this really enriches for kinase, not just based on their expression level, but it's biased to select for kinases an active conformation. So kinases can be on or off, and we wanted to look at the kinases and bias it toward ones that were actually on and potentially important for enacting signaling in these cells and in these models. And conversely, to look at the kinases where activity was lost. So we can see the targets of a particular inhibitor in these tumors after treatment with Brigatinib. And then we can use mass spectrometry to identify and quantify, and really the capture of these bound kinases is a function of their abundance, their activity state, and their affinity for the beads that we use. And so on the right, you can see a kind of a map, an overlay of this quantitative data, the kinome response. So we can indicate it, that's a phylogenetic tree of the human protein kinome. And so we can really represent it as the large and fold changes in kinase binding enacted by the inhibitors compared to a control treatment. And when we do this, or when we did this, I should say, in a meningioma cell line model after treatment with brigatinib, or in the genetically engineered mouse model, looking at tumor tissue after treatment with brigatinib, these are volcano plots. So everything you're seeing in the upper left, it's kind of scaled that way because everything was a loss of binding. So above that horizontal line are the significant kinases, and then the fold change represented on the x-axis. So you can see some clear commonalities between the two, between the meningioma model at left and the Schwannoma model at right. PTK2 or FAK, or focal adhesion kinase, and a couple of other kinases, GAK or cyclin G associated kinase. And in the mouse, I should say there are two colors because we collected tumor tissue and analyzed it after 3 or 7 days of treatment. And so this gives us a sense of the targets that were hit. These are all kinases, everything plotted here as a result of that proteomic assay. And so it gives us a suggestion of which kinases are affected, and it seems as if they may have lost activity based on our assay. They failed to bind. We could still detect them, but their binding was inhibited with brigatinib treatment. And another way we can approach this problem, kinase inhibitors exhibit polypharmacology, meaning even though brigatinib is advertised as an ALK inhibitor, we and others who have studied this drug know that it hits other kinases. And so we wanted to get a sense of whether the effects we were seeing here after 24 hours in the cell line or 3 or 7 days in the mouse model, we wanted to get a little more direct picture of what the targets of the drug were. And so the way we can do that is either a short or acute treatment. So at left, you're seeing the plot of the log2 fold change in binding with 2 different doses of brigatinib treated for 2 hours. So very short duration, less time for the phenotype, an arrested cell to really make itself evident, to get a better sense of direct targets. And then also we can make lysate. And in this case, at right, the plot is of lysate from the Schwannoma model tumor tissue. And again, I've labeled PTK2 or FAK1, the kind of name you're maybe more familiar with, popped out in both of these. Also with some additional targets that we went on to validate in kind of orthogonal assays to show they hadn't been published as brigatinib targets, but we were able to validate them by this assay and by some others. And I think it's important to bring up the issue that there are broad sweeping ways to study this. The Kuster lab, for one, did this with 240 kinase inhibitors and a mixture of lysate to see what targets are hit by drugs. But I think this really underscores the importance of looking at this specific model under study, under investigation. So we looked at schwannoma and meningioma after treatment with brigatinib. It really gave us some clues, since we saw efficacy, what kinases is it hitting, to give us a better hint at really the pathways involved and the potential vulnerabilities. And interestingly, brigatinib is an ALK inhibitor, but in all these models, there was no significant expression of ALK detected by RNA sequencing or by immunoblot. So it's pretty clear these other targets that are getting hit are likely responsible for the phenotype and the efficacy in vivo. So in summary, I just want to say and acknowledge the team, which I've got a slide showing everybody in a second. This systems biology approach really helped identify potential treatment options, indeed even moving a molecule into a clinical trial. It helped us identify the direct and relevant targets using this MIB-MS kinome profiling. We've got a few of the kinases listed there that really can prompt further study, genetic validation, or therapeutic refinement. If we can identify the key kinases and maybe get a cleaner drug to eliminate some of the toxicities potentially associated with the drug. And then lastly, Scott Plotkin is sponsored a clinical trial which is underway to investigate the potential use of brigatinib in NF2 deficient tumors. And lastly, I want to acknowledge this full team. I was in Gary Johnson's lab at UNC, but you can see in red are the members of the Synotis for NF2 consortium listed here, and then all of the additional co-authors of a lot of the data I presented. And thank you very much, and be happy to take questions. Oh, sorry.
Hi, Steve. I think you're hidden behind something that is blocking your camera, so we don't really see you. We all know you. Yep.
I am so sorry. I had flipped the thing. I apologize. That was terrible.
Yeah, I don't see any specific questions in the chat so far, so maybe we can move over to the panel. Just give it one more minute. Are there any questions? No?
Yeah, I wonder if I could ask something. So you identified a FAC in your MIBS assay, and Brigatinib will hit that, but have you tried a more specific just FAC inhibitor, since there's a lot of them around, and ask, would that give you the same efficacy without the other off targets?
Yeah, that's a great question. I should acknowledge the Kissel laboratory had found Crizotinib and kind of shown that it hit FAC also. So that's a few lines of evidence in mesothelioma studies where Merlin deficiency, predicted sensitivity to FAC inhibition. So yeah, those are great questions. We're kind of approaching them, trying, CRISPR and trying now more selective like defactinib that are on the market. Results are pending, but yeah, it'll be interesting. That's a great question, because if we get a really specific FAC inhibitor, will that help, or is it because it's hitting some other kinases?
Right. I mean, sometimes there's a virtue in polypharmacy, and maybe this is that case, but we'll have to see.
Yeah, it's a good question.
Steve, there is one other question from Barbara Franklin. Is there a list of plan B, C, or D options based on this approach? You can do the whole alphabet, right? What is on that?
Yeah, I think that compound library was 2,000 compounds. I think we are doing there are 700 kinases, so we're doing CRISPR kinome screens, so we're trying to broaden that. And that, I should have said, that panel, those were drugs where the mechanism was known, so they weren't necessarily all kinase inhibitors. So I think, yeah, approaches like this, to find potentially important molecules, and if they're targetable, there are larger libraries that could be screened. I think the system is good, though, and we happened upon one that seemingly worked in both meningioma and schwannoma, but I think, as others have mentioned, and maybe ependymomas, it could be as you get more and more specific about a tumor type, the vulnerabilities may not overlap, and they may be more efficacious if you did that kind of screening. But yeah, we're continuing this approach, looking to kind of rapidly advance to the clinic. We prioritize. We're very interested in this idea of drug repurposing. My lab in particular focuses on kinases, but we can see how other inhibitors affect the kinome, because it could be, and others brought this up too, that a combination therapy is going to be the most effective.
So, Steve, one question from Bart van Hazenbroek. In fact, two questions in one: whether you have tested AKT or PI3K inhibitors.
Yeah, that MK2206 in the meningioma model came out in combination with Brigatinib, and that data I showed that kind of by itself, it didn't really do much more than Brigatinib alone. And I think the clinicians could better speak to this. My sense is that those can be difficult in the clinic with the toxicities associated with PI3 kinase inhibitors, with the effect on insulin and getting insulin sensitivity, and AKT inhibitors, I think a bit more toxicity. I know in Wade Clapp's mouse model, I think they've tried, in the schwannoma model at least, tried AKT inhibitor, but there's some potential interest, I know, even further downstream in mTOR inhibitors. I think all these things are not necessarily being pursued directly by me, but others are investigating, and we can look at the effect on the kinome in those systems.
Right. Thanks so much. I think there is a lot of enthusiasm about looking at different targets, and AKT, PI3K, AXL. But I think we should move over to the next panel. But definitely, thanks so much, Steve. I think this is a very exciting future for improved drugs for NF2.